grandma has this dress.
No, neither.
I feel like Harry Potter. Too librarian. Too Bono?
No.
No. No. Big no.
Yes.
I look a little bit like a bumblebee. How long you been in these parts?
Blah, if sadness had a color. I bought this? Your Honor, I object.
No. This is so much like my dad.
That is the ticket.
Support for KQED Live comes from Berkeley Rep.
Disproportionate impact of
more than half of Black business owners.
And disproportionate.
Somehow we always find a way to rise.
To the blueprint builders, to the backbones of every block, for the curators of the culture, and for generations to follow. You might fall, but never fail. Keep rising, keep rising, keep rising. Apply for business, marketing, and tech makeovers on us.
So glad I'm flying out of Oakland today. Let's count the reasons why. For one, flying locally reduces my carbon footprint. Plus, my airport supports over 17,000 jobs in the East Bay. And it makes sense. The more I fly from OAK, the more flights airlines will add out of OAK. All good.
No matter where you live in the Bay Area, there are many great reasons to pick OAK and fly the East Bay way.
What you do with that extra hour is up to you.
Hi, everybody, and welcome to KQED Live here at the Commons. My name is Ryan Davis. I'm the Executive Director of KQED Live, and I want to welcome you all to this space. And I want to begin by acknowledging that KQED's headquarters are located on the unceded territory of the Ramaytush Ohlone people. We recognize that we benefit from living and working on this occupied land, and that we owe a collective debt to its original stewards, and that we wish to honor those Indigenous peoples that are still connected to this land. Now, for those of you joining us for the first time, KQED is Northern California's PBS and NPR member station, and KQED Live is our new multi-platform live events program that produces screenings and talks, performances, food tastings, and so much more that makes KQED's mission to inform, inspire, and involve tangible. And you can learn more about everything that we do and get tickets at kqed.org/live. And we want to thank our season sponsors that make KQED Live possible. They are Asian Art Museum, Berkeley Rep, Comcast Business, and Oakland International Airport. We're very grateful to them for their commitment to supporting our mission for civic and cultural engagement through programming like this. And now, I'm pleased to welcome to the stage our host tonight, the Executive Director of Science Friday, Danielle Danna.
Thank you. Hello. Hello. Nice to see all of you. Nice to be here in person. Thank you very much, Ryan. Thank you to KQED and the Deep Look team for welcoming us here this evening. My name is Danielle Danna. I'm the Executive Director of Science Friday. If you're not familiar with Science Friday, we like to think of ourselves as a one-stop shop for all things science news. But we are known for the public radio program, which some of you are likely familiar with. You can hear us on, you guessed it, Friday. You can hear us at 11 a.m. actually on KQED. If you get a chance to tune in, I would recommend it. You can also go to our website at any time, sciencefriday.com, and check out all of the materials that we have online, or you can also listen live during the live broadcast. Once again, thank you to KQED and the Deep Look team. Thank you to all of you for being here in person on such a lovely night in San Francisco and while the Warriors are playing. So I'm super grateful that you're taking the time to spend with us. I want to remind all of you to submit questions. You should have received cards. We want this to be a dialogue like any Science Friday program. So share your questions. They will pop up on the screen. Also for all of you online, welcome as well. It's really great to have you with us. You can also share your questions by putting them in the chat. The talented team at the Commons is here to receive those and feed them back to me. So without further ado, we're here to celebrate all things tentacled and these amazing invertebrates. Science Friday's been celebrating Cephalopod Week for years now, and I owe a debt of gratitude, all of you do, Science Friday does, to Diana Montano, our experiences manager, for putting together these cephalopod movie nights for years now. She does an excellent job, and they are happening all around the country, including tonight in Connecticut, which is where she is. So what we're going to be doing is we're going to be celebrating octopuses, cuttlefish, squid, all of their underwater relatives alive and extinct. And we have some experts here today to kind of share in that experience with us so we can ask some questions. Once again, make sure that you're sending those questions this way. Great. So before we kind of jump into the first film, I'll just let you know who we're going to have on stage with us. We're going to have Rich Ross, who is a cephalopod husbandry expert, Danna Staaf, who is a squid scientist and science writer, and a local celebrity, Josh Cassidy, who is lead producer and cinematographer for KQED's Deep Look series. So it's a good group. Are we ready to start the filming of cephalopods? Okay. Our first film is Cephalopod Inc.
We probably have the largest collection of cephalopods out of any research laboratory in the world, and we're growing extremely rapidly, which is sort of the nature of cephalopods. We probably have, I would guess, on the upwards of around 2,000 or 3,000 cephalopods in our collection. We're breeding them and we're expanding and scaling up to support our various research communities. So our program is very much like a startup. We're trying to get users to come back and use our animals, but our end game is not for economic profit. Our end game is for knowledge and advancing our understanding of not only cephalopods, but ourselves. My name is Brett Grassi. I am the manager of cephalopod operations at the Marine Biological Laboratory. So this is a new resource that's new to the MBL and new to a lot of research communities out there. Previously, models like zebrafish, frog, mouse, etc. have been utilized for decades and decades. Cephalopods are sort of the new kids on the block. We're really looking to try to promote this group of animals so that those scientists who have been working on the same model for decades may be able to ask the same questions of a different animal and see if they get different answers. There's no other invertebrates on the planet that can do what these animals can do. Not only do they have these immense abilities to change the color and texture of their skin, but when you look upon a deeper level, they're very unique and have a ton of advantages that we haven't been able to look at using other models. They've got 3 hearts, mini brains at the base of each of their arms, capable of movements on their own, abilities to edit their own RNA, very complex cognition. And so for all of those reasons, there's a lot of uses that cephalopods provide that other animals may not have been useful for. So the 5 species candidates that we've selected, on average, their lifespans are about 6 months or less, and most are reproductively mature at around 3 or 4 months. So we can effectively breed these animals very rapidly through multiple generations in a short period of time and really get a lot of questions answered very quickly. Because of the rapid metabolism, their sensitivity, and their feeding cues and behavioral cues are unlike any other animal. It's been kind of a slow road for the cephalopods to kind of make it to the larger research communities. And now through some trial and error, some research and development, we've been able to really advance our ability to not only effectively hold these animals, but keep them very happy under our care. Because we're sort of a startup operation here at the MBL, our day-to-day tasks are sort of all over the place. I manage the program, and we have Taylor Sackmar, who's really on the ground floor doing a lot of the animal care, the maintenance, the moves. For fish, you may need to sprinkle some flake food in once a day. For cephalopods, some of these species we're feeding 6 times a day. You can imagine that there is very drastic associated growth with that, so these animals are growing rapidly.
The basics are, you're always staring at these animals, how they're interacting. Are they getting too crowded in their tanks?
And so we are able to watch their skin patterns, watch that communication, and change sort of our animal care based on those observations.
The way we know a happy cephalopod is happy is just looking at different behavioral cues. So let's say for flamboyant cuttlefish, happy cephalopod for that would be settled down on the ground, and you can see them walking around, exploring their habitat. Whereas our pajama squid, happy cephalopod would be one buried in, because since they're nocturnal, during the day we just want to see them buried into that sand.
Every species is different, and it's that attention to detail and that focus that makes us successful when we're looking at our larger breeding program. We have certain species that we can hold in large quantities altogether, and that works really well because then they sort of breed with themselves, they have their own courtship displays, they can mate as they please. And then there's other species that don't get along in large communities. So for some of those species, we need to get creative. One method we've played around with is to introduce a barrier in between 2 octopus in the same tank. And that barrier has holes in it that are large enough for the octopus to reach their arms through in order to effectively mate. We really look at every species as unique, not only with their hatchling care, but with their egg care. Some eggs, when they're laid, are kind of left on their own. Eggs like that don't really take a lot of care, so when we harvest those eggs, we can put them in a cradle, something that just has some sort of good water flow. Certain species like the flamboyant cuttlefish and octopus will push jets of water over their eggs in order to promote oxygenation of their eggs and to prevent animals from settling on the outside of the egg surface. And so we do that with a soda bottle incubator that I developed long ago at the Monterey Bay Aquarium about 7 or 8 years ago. We have a bunch of scientists and researchers from all over the world that come to the Marine Biological Laboratory in order to use these types of resources we have available.
So a lot of our collaboration with our researchers is a lot of microscopy, a lot of embryology, a lot of genetic work with injections.
We're also creating innovative products in order to support the various scientific communities.
Like our IR dens to actually monitor egg laying.
It looks pitch black, but if you look at it through infrared light, it completely disappears. So at night we can have our pajama squids going around mating, laying eggs, and we can more effectively watch them and see what they're doing underneath those laying dens.
And we need to know when those eggs are laid, just so we have a better idea when the first cell division in the embryo occurs, and then we could actually get that egg over to the researchers in time.
We lovingly call our operations the Cephalopod Empire. It's not saying that other people in other parts of the world shouldn't be doing cephalopod research. The opposite. We want to encourage that growth, and we want to encourage the study of cephalopods. And that's because what we're really trying to do is build the personnel, the people around us, the people who are working with genetics, behavior, regeneration of limbs, and we can really rise together and learn more about these animals.
Thank you. I hope that you enjoyed that film. It is one of my favorites and a perfect way to kick off our first evening of Cephalopod Week. So without further ado, let's bring on our special guest. He has been raising and caring for octopuses for more than 15 years at his secret home lab and was formerly senior biologist at the Steinhardt Aquarium at Cal Academy. Please welcome cephalopod husbandry expert Rich Ross.
Hi, everyone.
Welcome.
Thank you.
She is a squid scientist, science writer, and author of Monarchs of the Sea, the extraordinary 500 million year history of cephalopods, and a forthcoming book called The Lady and the Octopus, How Genevieve Prue Power Invented Aquariums and Revolutionized Marine Biology. Please welcome Danna Staaf. And joining us from KQED's Deep Look team, they explore all of the tiny things in life that we love learning about so much. Everything from octopus suckers to flesh-eating beetles. The lead producer and cinematographer for the series, Josh Cassidy. Okay, great. We're going to kick this off. So once again, please inviting questions, so start thinking of them. Josh, the first question's for you. So we know that you get to explore all kinds of amazing things, but we're here to talk about cephalopods. So in the filming that you've done for cephalopods, how do you decide what to cover?
The whole kind of concept behind Deep Look is that it's a wildlife video series where you zoom in to find the answer. So it's either going to be about some tiny creature, like a fleas or something like that, or it'll be some small aspect of a larger animal, like how the texture on owl feathers allow them to fly quietly, or the microscopic barbs on a porcupine's quill that make it like stick in and not come out. So we're about to see a couple of episodes of Deep Look about cephalopods, but for me, my favorite one to film anyway, was about the chromatophores, about how they change color. So it'd be about, a pretty decent sized, squid, but really we're like zooming in to one little cell to see how they change color.
Yeah, there are some great videos coming up, and we'll get into that, and I know we have an audience question about that. Rich, you have been living with cephalopods for some time now. What have you learned? What can you share with the budding ceph heads out there that are thinking about maybe doing this at home?
Get good at keeping saltwater animals before you start with cephalopods. Because my thought is the whole trick to keeping them alive and happy is, once I did a talk about some baby cephalopod that we were trying to raise, and somebody said, why don't you just give them what they need? I was like, I'm trying. We don't know what they need. So you got to get all the bases covered that you know you can cover, and then try to figure out what they need beyond that. And saltwater keeping is an art unto itself. So if you want to keep cephalopods, learn how to keep saltwater fish, then learn how to keep saltwater coral, then think about cephalopods.
Well, you just gave me a really nice intro to my next question for you, which is that I have heard you are now, or have been, but we're now talking about you're doing coral spawning?
Yeah, I've been doing saltwater since I was a kid. So cephalopods and corals, all of that's been going on for 30 or 40 years. And one of the new frontiers on corals is spawning corals in captivity and raising them up. I think we were the first lab to do it in America, and maybe the second or third in the world, so that's pretty great. And it's funny because you go with coral and cephalopods, and cephalopods are fast, they eat a lot, and they try to escape all the time. And coral is essentially a rock. So they need the same water quality, but everything else that goes into the tank is very different.
Yeah.
So yeah, spawning coral is amazing because it's about 8 months of hoping as they develop their gametes, and then it's about 3 months of insane amount of work keeping the babies clear of algae and things like that.
Well, you're lucky family. You've brought all of this home.
Yeah, they are very good to me, my family. They even came here, so that's even more exciting.
Danna, tell us a little bit about your research and studying squids in the lab.
Sure. So I have to go back in time because of what Rich said and admit to Rich and to the public that the very first saltwater aquarium that I kept was for the purpose of keeping an octopus. I did not take your future advice that I would hear because I fell in love with octopuses as a kid, and so I was insistent that I needed a pet octopus, and I got my dad to get in on it with me, and we got a saltwater aquarium so that I could keep a pet octopus. And so then by the time I was all the way through college and looking at graduate school and going to get a PhD, I thought I still had this fascination with octopuses going on, but I ended up in a squid lab. And so I actually had to broaden my horizons a little bit, just a little bit. And I ended up spending 6 years studying squid, which are different from octopuses and similar to them. And the whole group cephalopods, they all do a lot of the cool things that we saw, the color changing and these really interesting life cycles. And so I actually studied squid sex in babies, was this short version of my very long dissertation. Just 4 words. And I was studying the species called the Humboldt squid that gets, as an adult, about as big as me, which is not very big, but it is large for a squid. And the cool thing about them, one of the cool things, was that the babies were teeny tiny. So they were hatching out of their eggs as like little grains of rice and then growing to that big size in like less than a year.
Yeah, amazing.
And so that was a lot of what I was trying to do, is what are they eating? How are they growing that fast? How are they surviving, basically?
Yeah. Well, that kind of brings me to an interesting question. So we talk a lot about the visuals of cephalopods, looking at them, all the camouflage, and how do you write about something that people comment all the time on, like how it looks? And
right, like naturally they should be movie stars, and yet I try to put them in books. So like clearly I'm in the wrong field, but it seems to work out all right. We do put some illustrations in the book, and I think that one of the things that makes them both really fun to write about and hopefully appealing as well is that there's so many good stories about them. And the way that they change color is one of those stories. But it's a story of like deep evolutionary time that I ended up getting to tell in one of my books that I'll have available, which is just a little plug there. But it goes all the way back to these animals having evolved from living in shells when they didn't need any camouflage, and you couldn't even see their skin. And then over hundreds of millions of years, as fish evolved, as whales evolved, they had to evolve to compete and escape from those predators, and we ended up with the squid and octopuses that we know today that have no shells or just very tiny internal shells, and then this amazing camouflage. So I think that's what I tend to do, is I look for the stories that you can tell even if I don't have the visuals.
Yeah, that's great. And I'll just do another plug that Danna will be out in the lobby afterwards signing books and has them available, so don't hesitate to join us out there afterwards. So before we kind of get into the weeds, we're going to jump into our next two films, one by Deep Look and one by Science Friday. And we're basically going to be investigating octopus arms and mobility. So without further ado, let's jump in.
They can crawl. They can squeeze and hide. They can even use jet propulsion. And just when you think the octopuses couldn't get any more versatile, they up and walk away. And while it's utterly absurd to look at, once we stop laughing, maybe we should ask, why would one of the most agile creatures in the ocean choose to scamper backwards? That momentous question captured the attention of Dr. Chrissy Hufford, a senior researcher at the Monterey Bay Research Institute whose graduate studies at UC Berkeley focused on cephalopod behavior.
Generally, I try to understand how octopuses survive in their everyday lives, avoid being eaten, find mates, and find prey.
Documenting the survival strategies of octopuses isn't easy. You've got to be willing to spend a lot of time on remote tropical beaches.
My job is a lot of fun. I'd wake up at dawn every morning, get in the water, and get out for a brief snack at low tide, and then get back in until sunset.
It's hard work, but somebody's got to do it.
Unlike other octopus studies that had been done, I was actually able to follow individuals all day long on snorkel.
Particularly the cryptic algae octopus.
The algae octopus tends to come out during times of day and tides and in certain habitats that mean they tend to clump in the wild. And there are lots of individuals that use the same area and prefer that same area, so they're bumping into each other, mating, fighting, competing for similar resources. And I could watch this happen in real time and pretending I wasn't there.
Although on occasions, her subjects realized they were being observed.
I'm assuming that they considered me a predator in those cases. Octopus's main defense is not to be seen in the first place. That's their best chance of not being eaten.
And if all else fails, they can just jet away.
And in that shape, camouflage isn't even a possibility.
It was in one of these defensive moments that Dr. Hufford witnessed an entirely novel attempted escape.
I was working with a film crew for a documentary series called The Shape of Life. It was easy to get them to act defensively when you're chasing them with a camera, and we got footage of the coconut octopus walking on two arms. It was hysterical to see. We laughed and our masks filled up a little bit with water. Then later I saw the behavior in the algae octopus. We again laughed at how hysterical it looked, but also we understood the biomechanics significance of it. Octopuses have very strong arms, even though they don't have bones. They have 3 bands of muscles that oppose against each other, and the volume of those muscles and their arms stays constant as it changes shape. That's called a hydrostatic skeleton that allows them to use the internal pressure of their arms for support.
But just because an octopus is physiologically capable of walking backwards doesn't make it the best form of escape. Or does it?
When octopuses jet to escape a predator, there's so much internal pressure that's built up inside of their mantle cavity or inside of their body sac that it actually stops their hearts briefly.
Going into cardiac arrest while swimming away in a panic probably isn't ideal.
Walking backwards allows them to use 2 of their arms for locomotion and 6 of their arms for camouflage or to otherwise make their shape seem unrecognizable.
And while only a few species thus far have been documented fleeing on foot, or rather arms, Dr. Hufford believes that this escape plan isn't a difficult trick to pull off.
Just the way We don't have to stop and think, move left leg, move right leg. It's a type of motion called feed-forward movement where it's almost automatic once it gets going. And I think it was part of their normal locomotion repertoire of how they can move around.
Get another method for these skittish invertebrates to show off having no backbone. For Science Friday, I'm Luke Groskin.
This octopus is making a quick escape with the help of its suckers. The suckers are incredibly strong. And while we only have 2 hands to grab things, an octopus holds on with hundreds of suckers running up and down each arm. Suckers come in pretty handy to get around or to grab a bite to eat. Or to hang on to your keeper's arm. So how do suckers stick so well? It's not super glue, it's water pressure. Each sucker has a ring called an infundibulum. It creates a watertight seal. Inside the sucker, there's a chamber, like a cup, called the acetabulum. It's full of water. When the octopus expands the chamber, it lowers the pressure inside. The higher pressure outside pushes against the sucker, creating a powerful grip. Suckers get such a workout that their skin sloughs off every few weeks. See that ring-shaped piece of skin floating by? It used to be the bottom of a sucker. Suckers have another superpower. They actually can smell and taste. That helps the octopus tell there's no snail inside this shell. Hundreds of suckers tasting and smelling and flexible arms moving every which way create a huge amount of information. A lot for the octopus's central brain to process. So each arm has a mind of its own. In fact, 2/3 of an octopus's neurons are in its arms. That means the central brain isn't really calling all the shots. The central brain tells the arms how fast to move, but the instructions on how to reach come from each arm. It's a nifty way to process all that information, making the octopus the well-choreographed creature we know.
That was amazing. Josh, I'm going to jump right in. Did we see your arm in the video?
No, I have to be behind the camera, so I don't get bit by things and such on the show, or in this case, caressed by the octopus. Yeah, I've tried a couple times to like film and be in front of the camera at the same time. It doesn't work
Very
well.
difficult.
But
you don't
actually,
have 8 arms.
The first shot you see of the day octopus coming out, that's actually Rich.
My big puppy mitts.
Yeah, and you could actually, you didn't see it in there, but from behind the scenes you could see on sort of an image that sticks with me, he had taken the lid off for us to film it, and it sort of came out like the Kraken, you know? And then Rich was sort of encouraging it back into the tank, and it grabbed a hold of his arm, and I could see like the arms like wrapping around on his tattoo of an octopus on his arm. And it's like, yeah, it's something special.
So what can you tell me about the experience that maybe members of our audience have not experienced themselves?
Well, first, just that sound. It really just sounds like a million little wine bottles opening, like that sort of pop. And then also, for me, I really like the look in the octopus's eye, kind of like a little critical, like, not so sure, you know? It almost like it has like eyebrows. I mean, it doesn't, but it looks like it. And then I was surprised by how much the color changes a lot, but also the texture of their skin changes. Yeah. And they have control over that. I think that's super cool.
Yeah. Quick follow-up. On your next occasion to film octopus, are you going to stick your arm in the tank?
I guess I have to now.
Yeah, I feel like you do. I'm going to take an audience question and kind of go back to something that we talked about a little bit earlier. But we have a question from Gemma about pet squids or pet octopus. Is this a thing?
I could jump in on that. So it's a really good question. Do people have pet squids or pet octopuses still easier? Pet octopuses are definitely still easier. If you have been to any aquarium, the most likely cephalopod that you're going to see is going to be an octopus. Even the tentacles exhibit at the Monterey Bay Aquarium that's chock full of cephalopods has very few squid. And really the squid that are on display there are unusual squid species because the typical squid lives in the open ocean. It doesn't really associate with the bottom. It doesn't associate with coral or rocks or anything. It doesn't have a den the way an octopus does. And the way I think about it is actually an octopus, for all that it's like a really alien thing, is also a lot like a human. It has a home. It often likes, most species, they like to come back to their home. They live mostly on 2 dimensions. They're walking around a lot more than they're swimming. And they navigate the way we navigate in our neighborhoods, whereas a squid is living like way out in the open ocean. We don't really understand how it lives in this 3-dimensional environment. And it's really hard to replicate in the lab. And so actually when I was working on the Humboldt squid, I was working right next to the Monterey Bay Aquarium at the Hopkins Marine Station, and we were cooperating with the aquarium to try to see if we could keep Humboldt squid alive. And we actually had a couple of big Humboldt squid in big tanks, not on display anywhere. It was all behind the scenes. And it was impossible. I mean, they swim too fast, they bump into the sides, they rub their very delicate skin. So anyways, I get sort of excited about it because it's so frustrating because it would be so cool to keep them in captivity, but we can't do it yet.
Yeah. Well, Rich, as somebody that has worked in an aquarium and has cephalopods at home, what's the difference basically between an aquarium and a home lab in your experience?
In a home lab, I can make decisions instantly and buy anything I decide that I need to have.
Okay, maybe for the cephalopods.
For the cephalopods itself. Part of the difference is there's less traffic in the home lab. So there's less people walking through and seeing the animals. So like when we collected some day octopus to put on display, it was probably one of the ones you filmed, in Hawaii, and we brought them back and we knew they're gonna go on display. And they're very skittish when they come in. They don't know what any of this stuff is, and people are looking at the tank and banging on it. And so how do you get them used to that? And so I would take iPads and I would put them in front of the tank or a little bit behind the tank, by the glass, so the octopus could watch it. And I would show it movies and Christopher Hitchens debates and anything that had people, chest up, because that's what the animal's going to see when it looks out of the glass, or in little kids going like this.
Get it ready.
Yeah, and I know Brett took that Brett Grassi, when he was at Monterey, he took it a step further where they actually went to one of the tanks and they put a camera in the tank looking out of the tank and used that film to show the animals before they would go on display, so they would really see what's going on. I think they did that with squids too, because squid are a bit of a nightmare.
Yeah. Well, let's see here. We talked a little bit about your spawning coral, and we do have an audience question about the hardest part of spawning coral.
The hardest part of spawning coral is everything we know about spawning coral now, and even developing gametes, is based on lunar and solar cues. And so it takes them about 8 months to develop the gametes, and then about 3 months for those gametes to mature and then spawn. And all of that is cued.
Gametes are eggs and sperm, right?
Yes, they are. Thank you for that. And getting them to, so it's light pollution seems to play a really terrible role in making them not develop or not spawn. So basically what we do is we put them in a dark room and let a computer do all the lunar cycles and sun cycles and intensity cycles and heat cycles, and that's a real pain because you've got to go in and out of a dark room to do any maintenance on them. So that's where we are right now. That's what we think we have to do, and so that's what we do. So that's the hardest part.
Question for you, Danna. I heard that during the pandemic you did some sidewalk science Q&As, which I'm sad that I missed, and I'm sure other people here are sad that they missed. Can you, first of all, tell me if you got any questions about cephalopods in one of these Q&As, but also kind of talk to me a little bit about like some of your favorite or unexpected questions that you got while you were out there.
Absolutely. It was really fun. So I don't know, it feels like a lifetime ago, but you may recall that a lot of people in the first few months of lockdown were where we couldn't really go very far away from our homes, got really into sidewalk art. Certainly our neighborhood did, and so there were kids, writing encouraging messages out on the sidewalk, and I just had this. I had actually been teaching science to my kids' classes at school, and so school shut down. I wasn't doing that anymore, and I just had this thought of like I could do that to the neighborhood, inflict it on the neighborhood, do it for the neighborhood. Yeah. And so I took our chalk that the kids had been playing with, and I wrote on the sidewalk in front of our house, "A squid scientist lives here." And then I left the chalk in a bowl next to that, and with some little Q's and A's on the sidewalk. And I didn't really know what would happen, but people got into it. And so it got so that my kids, the first thing in the morning, would rush out. Like the early dog walking crowd had left questions, and they would come and be like, "There's a new question, Mom." And so I got quite a lot of questions, and they were everything, like these sorts of questions that people are asking. What's your favorite cephalopod, or what's the biggest one, or kind of out of the blue, like, do squid have an alpha male in their social groups? It's like, that's no, but that's an interesting question. And then my favorite one was probably the person who wrote and put a little line on the sidewalk and said, if this were the end of a giant squid, where would the head be?
Nice.
And I was like, I can't believe I didn't think of doing that already. And so, of course, I drew it out on the sidewalk, pasted out the actual length, and it went way out in front of our neighbor's house. Because I couldn't go all the way. I went to the head, but then I was like, we need the arms and the tentacles, so I kept going.
That's amazing. Thank you for doing that. Such a public service. So we have talked about writing and cephalopods, and we have got a great audience question, and I'm going to direct it to you, Danna, as the writer, even though I know that Rich probably has opinions about this, but Dracoloid has asked, can people get attacked by a cephalopod? And what does that look like?
I am particularly well equipped to answer this question. By having spent 6 years studying a species that many people know as one of the most aggressive cephalopods out there, the Humboldt squid. Some people called them red devils. There were people saying that the name for them in Mexico, where they're very common, is Diablo Rojo, but I actually never heard anybody call them that when we would go to Mexico. They just call them calamar gigante, giant squid, or very large squid. And they are very aggressive predators. They will attack at each other. That was the thing that probably the scariest thing that I saw them do is if you were fishing for one, you had to pull it up really fast or you'd only get part of one because the other squid had come in. But there aren't really any accounts of people being attacked by them in any sort of usual encounter. And they are generally going for prey that's much smaller than us. They tend to go after little fish and shrimp and stuff like that. And then I suppose since we're talking about anybody being attacked by cephalopod, we should briefly mention blue-ringed octopuses because they are the only cephalopod that has caused documented human fatalities. And they're very tiny, but they're very poisonous. So, and they don't go after anybody. They'd much rather hide. Like Chrissy was saying, their first line of defense is not to be seen, but if somebody does really mess with a blue-ringed octopus, it can bite them. And
Fantastic. We've got some good questions here, so I'm going to jump in and kind of answer them. I kind of want to save this one for later, so I'm going to. All right, let's start. How do octopuses produce ink? Thank you, Elliot.
How do they produce ink? Comes out of a gland and squirt it out.
They do. Yeah. They have an ink sac, a special organ. It's mostly melanin and some mucus.
Okay. Yeah, great.
There's a lot of mucus in cephalopods.
It's a lot of slimy stuff.
I mean, if you ever get the opportunity to touch one, which I don't necessarily advise, we've all seen some accidents online, but they are generally very slimy. Yeah. Okay, I love this question because I love them, but why don't the nautilus get more love?
I drew this. It's not biologically accurate, though, because nautiluses don't actually care for their young. I just wanted to draw it. So, but the nautiluses, in some ways, get too much love, right? Because they're one of the cephalopods. They're maybe one of the only endangered species of cephalopods. Are there others?
It's hard to tell, but yeah.
Yeah, because their shells are so beautiful. So nautiluses are the only cephalopods with external shells still. I talked a little bit about how most of them have evolved away from that. And the shells are so beautiful that they're collector's items. And so people have collected too many. And but just in the last few years, nautiluses are protected now by CITES and by the Endangered Species Act. So they're getting some more love, hopefully. Yeah,
they're also hard to keep, hard to display, and boring.
Yes, I was going to say.
Bad at swimming.
I mean, yeah, they're terrible at swimming.
Yes, very slow.
Knocking the tink, 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 tink for hours.
Yeah.
They're very interesting for about 4 minutes a day, and then they're less interesting.
Yeah. Well, with that, I'm going to ask the second part of this question. Do each one of you have a favorite cephalopod or an obscure cephalopod you'd like to tell us about?
I was surprised by how much charisma cuttlefish have. They really seemed like they would, if I saw them in the tank, they didn't seem like they were as shy, and maybe you know better, but yeah, they just seemed kind of adorable.
Yeah, that's a good answer.
Octopus cool and Squid, I wouldn't call them adorable though.
I would.
Okay.
Absolutely.
I mean, yeah, they are, but
I mean,
wrong,
super
but
cool. Super cool. Adorable.
But cuttlefish.
Yeah, okay, maybe.
Favorite? Oh man, that's such a tough question. They're all pretty cool. The ones I love the most, I guess, are I got 5 are going to be the dwarf cuttle, because that's the first one I work with and close the life cycle on, so that's really exciting. Then there's the flamboyant cuttle, which is just insane, and I love that animal. And then the larger Pacific octopus and the lesser striped Pacific octopus, this one here, I did a bunch of work on, and they do all kinds of weird stuff. And then Argonauts are just, Argonauts would be the next one I'd really, really like to work with.
I think Danna feels similarly, but
go
ahead, Danna.
Can I ask the audience?
Oh.
If they have a favorite?
Yeah, just like show of hands thing.
Yeah, of
Of
course.
Because I heard some clapping for cuttlefish, so like you can talk to each other after. Whose favorite cephalopod is a cuttlefish? All right, so take note of each other. Whose is octopuses?
Cheating.
And Squid? Yes! And some of you probably have specific species, so you should talk among each other afterwards. Nautiluses, though? Any favorite Nautilus favorites? Oh, good. Very nice. All right. Thank you for humoring me, and whoever turned the lights on. Bless you. We could talk about Argonauts, because they're awesome, but I have to confess that my favorite cephalopod, nobody will be able to guess why, is this miniature Californian octopus called the California Lilliput octopus. This is so tiny. I'm tiny.
I feel like there might be a deep look coming up. Yeah, right?
Yeah, they're really cool. They're the first one that I tried to study. So I think I have a similar, I was an undergrad and I was trying to study these and my advisor was like, nobody knows anything about them. And I was like, with the passion of the new scientist, I will be the one to find out. And what I found out was why nobody knows anything about them, which is that they're really hard to find. But they live in kelp in like the little root holdfast structures and they're really cute.
Yeah, I do feel like you have an idea here.
They're around here?
Yeah. Well, we could talk about this, I feel like, endlessly, and I've got some really great questions from our online audience as well. So without further ado, though, I think it's time for us to jump into skin and watch our next video.
Jump out of
skin. Into skin.
Cuttlefish, octopuses, and squid have an almost otherworldly ability to control their appearance. What makes it possible are these spots. They're called chromatophores. They're like tiny water balloons filled with colored pigment. When the balloons expand, you see more pigment, more color. When they contract, the color shrinks to a tiny dot. The overall effect can be really dramatic, and for good reason. These animals don't have protective external shells. They're unarmored, naked, and they aren't great swimmers either. Camouflage is their best defense. They have to be good at it. Octopuses can change their body position and the pattern on their skin to match rock or coral. Octopuses and cuttlefish can even change the texture of their skin to throw off predators, become bumpier and more rock-like. But squid often live in the open ocean. How do you blend in when there's nothing except water to blend into? They do it by changing the way light bounces off their skin. They can actually adjust how iridescent their skin is using light-reflecting cells called iridophores. They can mimic the way sunlight filters down from the surface, hide in plain sight. So how do they control all this color change? Is it voluntary or some kind of built-in reflex? That's what researchers at Stanford University wanted to know. So they anesthetized the squid and then snipped the nerve from the brain that controls the chromatophores, but only on one side of the animal. The brain essentially couldn't send messages to the tiny muscles that control those chromatophores anymore, almost like turning off a light switch. But after a few days, they noticed something strange. The chromatophores began blinking again, even though they were no longer getting signals from the brain. So what does this mean? Well, what it suggests is that color change might be a bit like breathing is for humans, something we can either choose to do or do automatically, only even cooler. Because unlike breathing, color change requires an awareness of your surroundings. And in these animals, that awareness is spread throughout the skin, as if the skin itself could see. It would be as if your skin knew what color the walls were, even with your eyes closed. For a soft and squishy creature trying to stay alive in a very big ocean, it's a pretty spectacular defense.
So I'm going to jump into an online question from Danny. When we're looking at these camouflage creatures, I imagine that they can get lost when you're watching them, Rich. Have you ever lost one? And do they ever try to escape?
Octopus try to escape all the time. I remember converting a tank from octopus to coral and pulling off the lid and throwing it away and going, yay! But yeah, they absolutely try to escape, and they're very easy to lose sight of. So you have to become very patient and learn to do this a lot.
It's like my job.
You just sit, you just sit there very still and watch for the little tiny bit of movement, and then you know it's still okay. You know, the people sometimes freak out and start trying to find it, and then you're in big trouble because it's going to hide even worse. You want it to feel comfortable so it comes out.
Have you lost one?
I have not lost one.
Okay.
No, I try to listen to everyone who said, be very careful. Yeah. When we were collecting them, we went on a trip to collect the Hawaiian day octopus, and the thing to do was, you know, we told everybody was, so you're all diving underwater. If you see one, you do this, and you just sit there like this and wait for other people to look at you. You point at it, keep your eye on it, and don't look away, because if you do this, it's gone. I mean, you literally will know where it was, and you won't be able to see it again, and it hasn't moved.
Yeah.
It just vanishes if you don't get that bit of motion. So,
wow.
Escape artist.
Yeah.
Great advice too. Danna, let's talk a little bit about the Argonaut, because you have a book coming out, right, later this year?
Yeah, in October. It's called The Lady and the Octopus. So it's sort of a two-parter. It's a book about Argonauts, which are just some of the absolute coolest octopuses out there. And this woman, Jeanne Villepreux-Power, who discovered a lot of what we know about them back in the early 1830s. And it kind of connects to what we're talking about with the aquariums, because what she did in order to study the Argonauts and figure out what they were doing was basically invent aquariums. She just came up with this idea for how you could keep an animal in your own home happy, that nobody had really tried to do before. Everybody was just studying dead specimens that they'd fished up and had preserved. And she was like, no, we need to know what they're doing, and we need to see them actually doing it. And the reason that Argonauts were such a good fit for this is that they are the only octopus that makes its own shell. So this gets back to the shells. We have the nautiluses that grow a shell. They grow a shell like a snail does. They have it from inside the egg, and they are attached to it. And if you try to pull a nautilus out of its shell, you either break the shell or you kill the nautilus. Argonauts are totally different. They look like regular octopuses for the most part, but they have two very strange arms that are these big spread out membranes, skin, very thin skin, and they actually use those membranes to secrete a shell that they can climb in and out of. They're not attached to it. And it's only the females that make the shell, and they make it as an egg case, but they make it throughout their lives. So even a very young female will have a tiny little shell cap, and then it gets bigger and bigger. And there was this debate for hundreds of years about whether they actually made it themselves, because this creation of it with the arms had never been observed. And so it wasn't until Jeanne Villepreux actually built these aquariums and watched them doing it in real time that she was able to convince everybody they really do make their own shells. Before that, a lot of people were like, they're like hermit crabs, they must steal the shell, they must find some dead snail to take it from. And she's like, no, they really do it.
They're so fantastic. Cool.
Yes.
For so long, we didn't know how they controlled buoyancy in the shell. This is an insane, they put air in it. They put a bubble in it.
And they go to the surface to get the bubble.
Yeah, and then they let it out, and they go sink down, or they go swim back up and get more air. And they ride jellyfish. Sorry, jellies. They should be jellies, not jellyfish, because they're not fish at all. So cuttles should also be cuddled, but I can't get any traction on that. But you got the jelly doing this, and the Argonaut will come and sit right here. And Helmut, when was trying to take pictures of them, every time he went around, the Argonaut would, to get a picture of it, the Argonaut would turn the jelly so he couldn't get a shot. And he had to get two other divers to come in to distract it, to turn it to be able to get a photograph of it.
I mean, it's basically tool use, right? Like, they're using the jelly as transportation and defense.
It's the original jet ski.
And taking a snack, too.
Yeah.
Because they'll take bites out of the jelly.
They do. Yeah, the tool use in cephs is a whole thing. That's your book after the next one.
Oh my gosh, that's fascinating. Well, you've also now just touched on like the difficulty that sometimes filmmakers and photographers have with trying to capture these creatures. So like, Josh, can you talk a little bit about like what it's like to try to film these chromatophores like in action? I mean, that's got to be really difficult.
It's the most fun part of the job as well, I have to say. But I had never seen this before. I knew that they changed colors, but most of these squid that I had seen were breaded and deep fried, like some marinara. And I must say they're way cooler when they're alive. They're also delicious. But anyway. And actually, I've seen, since filming this, first the researcher at Hopkins Marine Station, so it's Stanford's Marine Station, was studying exactly how they open and close these different chromatophores to change color, because they can open up, say, like all the brown ones, and then they'll turn that color, you know. So they're able to coordinate it. But what she was doing was putting a small section of the skin in a little like petri dish you saw, and putting a little voltage through it, and then trying different pharmaceutical drugs to figure out which ones were affecting. Yes. So it was like very focused, you know, research. But I had noticed after this that if you go to a fish shop that has fresh fish, and they're very fresh, you can actually sometimes see, because these are just market squid, you'll see them in the display case, and the chromatophores are like changing color. I know this is a horrible thing to say in front of you too, but I was very surprised that it keeps going for a while after they passed on. Yeah.
The first flamboyant cuttle I was able to work with, I got through a collector, and they sent it up to me, and it was not alive, but it was almost alive. With the skin changing colors. And so I spent 5 hours giving it CPR with a little turkey baster, hoping that I could jumpstart it. It didn't work, but
sad story.
they will continue changing color for a long time after they're not alive.
But so as far as filming it, there was they had a bunch of these squid in tanks, and I think those are sort of the coolest shots. And then also, several of the close-ups are under a microscope, so we have a camera, you can use an adapter and attach it onto the microscope and get those like extra super close-up shots.
Very cool, very cool. Well, let's jump into our next video. It features somebody you should be familiar with. We have a celebrity on stage. So without further ado.
This animal is the cephalopod lover's dream animal. It's Octopus churchii, or the lesser Pacific striped octopus. This guy's small, about the size of a quarter. You find these little octopus in the Pacific coast of Central America in the intertidal zone. So shallow water just off the beach. What makes this octopus so striking is it's got these kind of black and white or black and cream stripes. It's got chromatophores in its skin, and it doesn't get different colors really, but it'll change the density of the stripes. It's just kind of gorgeous. I don't know any other way to describe it, except when I look at it, I'm very happy. This is my secret home lab. This is where the magic happens. All this is now fish, or fish breeding, and I grow some coral in here now. And I got this rack specifically with the idea of breeding octopuses in mind. That's the larger Pacific striped octopus. I am studying this animal because I love this animal. This animal is one of the ones when I first started getting into cephalopods that I thought I would never, ever see alive. And I've got 6 of them over there right now. 2 females and 4 males. Telling the sex of cephalopods can be really difficult. The lesser Pacific striped octopus is actually very easy to tell if it's a male or a female. On the ends of their arms, the female has just suckers, but the male has this little kind of fuzzy papillae. And once you're able to spot the difference, they're actually very easy to sex, which makes your life as a cephalopod escort service, I guess. It makes it very easy. And it's on. When you're an octopus, having your lover eat you is always a possibility. Generally, how these guys mate is with the male as far away from the female as possible. The male octopus has a modified arm called a hectocotylized arm, and that actually goes inside the mantle. It's more complicated than that. You can see his arm just behind these bunches of arms. One arm is inside of her mantle cavity. The penis delivers the sperm down the arm through peristaltisis, like when you swallow. The groove in the arm kind of squeezes in and out and pushes the sperm packet forward. And then that gets to a thing on the tip of the hectocotylized arm called the ligula. And the ligula is what the octopus actually uses to place the sperm packet. Ooh, spermatophores. Wow. Did I mention octopuses are amazing and cool? They really are. There we go. And a nice exit. How long before the female lays an egg clutch? That's a hard question to answer. One of the things I'm hoping to find out when I have completely closed the life cycle is being able to answer that question. We know it's iteroporous, meaning it has multiple clutches of eggs, so it's not just one bunch of eggs and then they die like other octopuses do. They keep on going, which is a big deal. Almost no other octopuses do that. It's got large eggs, and in cephalopods, large eggs means big babies, and big babies generally means not a larval phase. There's not that many octopuses that hatch out as mini adults. These octopuses live for about a year to a year and a half. I'm not saddened by their short lives. If you've been doing this for 10 or 15 years like I have, you get used to that as a fact of life. It's actually kind of beautiful because it's our life, but just compressed down into a year. I think cephalopods in general are the coolest animals in the world, and I've got them in my house, and I'm doing useful work with them. What more could you ask?
Very sweet.
It makes me want to get right back into it.
Yeah.
Well, I was going to ask if that's something you're currently doing.
I had to take a break because I got kind of depressed and didn't have the wherewithal to take care of them in the way they should be taken care of. So I kind of stopped with them. And then the pandemic happened, and then everything's insane. But I got to tell you, I still have that system. And watching that makes me go, maybe just a little drink.
It is a good thing your family's here then.
Well, it's funny. Every time I talk about like taking the lab down or getting rid of a piece of equipment, she goes, no, you're going to find something that you're going to want to work, and you'll need it all. So she's very nice to me.
So like, tell me, I mean, it's been a few years since this was produced. What is happening right now? I mean, all three of you might be able to answer this, but like, what is happening right now? What's on the forefront or the frontier of cephalopod research? Like, what are we going to be talking about in a couple years? What is Josh going to be filming?
Rewriting your own DNA?
Yeah, maybe we'll all be part cephalopod by then.
Nice. Change color.
Yeah, that's the dream, right? I mean, I feel like all these videos, it just links so nicely together because this sort of loops back with trying to close the chair life cycle with the cephalopod empire that Brett Grassie's running, because they have that now, right? They've closed it, or they closed.
I don't want to take their thing.
But it's closer. So that particular species, I know that they've been working with a number of labs and like building it up as a model system. A model system is sort of like a lab rat or like a fruit fly or something. And the idea that there are cephalopod species that could really be raised that abundantly all around the world. And I think what a lot of people are really excited about is what you can do with them then genetically. Because at the same time, just general genetic techniques have come so far. Like it used to be such a big deal to sequence a genome, and we now have probably half a dozen cephalopod genomes, just completely sequenced. There was a relatively recent paper comparing all of them, showing, yes, they all do RNA editing. They all have this weirdly jumbled set of genes. They all have this really expanded set of genes called protocadherins that I learned how to pronounce because it was so exciting. Because we also have a bunch of protocadherins, we vertebrates. So cephalopods, which are invertebrates, have no backbones, and vertebrates both independently evolved big brains, and now we think that might be connected to these protocadherins. So
interesting.
There's a lot.
That was we did have an audience question about RNA horizontal transfer. So I don't know if anybody wants to dive in a little bit deeper.
That's all not me.
I don't know of any horizontal transfer specifically. That is a term that's usually used to describe when a species gets DNA usually from another species. So like, and it happens, we'll pick it up often from our symbionts. So like if you have bacteria living with you for millions of years, then you end up starting to share some genomes. And you have to stop me because I'm going to start telling a story about roly-polies. But I don't know of any examples. There's a good story about roly-polies. Ask me later. But I don't know of any examples in cephalopods. But whoever asked that question, tell us about it afterwards. I always want to hear it.
Yeah. Great, thank you.
I think we're going to find out that inside our brains are small cephalopods controlling us.
It's pulling the levers.
Little homuncular
Maybe just yours,
pods.
Rich.
Just me?
Would explain some things,
Rich, you talked at the beginning of this evening about, the sensitivities of the water and the environment that you have to maintain for cephalopods. Yeah. Can you talk a little bit, this is an audience question, about how temperature and oxygen conditions in a warming ocean might be influencing or impacting cephalopods, octopus, squid?
Yeah, it's going to impact everything. So if you're a warm water octopus, the water's going to get it's all sad now. It's going to get too warm, and you're going to have a problem. If you're a cooler water octopus, it's going to get warmer, and you're going to have to go deeper if you can do that. If you're a cold water octopus, you might be in trouble. So yeah, that's the temperature rise is definitely problematic. Even also the acidification is bad. So the nautilus, as it gets more acidic, the shell will start to dissolve. We've already seen this shown in corals and in problems in fish as well, and their otoliths. I think fish have a otolith.
Otoliths, yeah.
So yeah, it's going to affect everything. I don't think they'll disappear, but we may not have the plethora of wonderfulness we have now.
Yeah. On a sweeter topic, Danna, you told us that you are writing a book about baby animals. I must assume that this includes baby cephalopods. And we do have audience question from Taryn about what do baby octopuses eat. So I'm wondering if you can tell us a little bit about the book, Baby Animals, and then answer Taryn's question.
I can definitely do that. I see Rich shaking his head. He's like, they don't eat anything. It's hard to raise them, that's for sure. But yeah, so I have a book about baby animals that, yes, the idea is that everybody will say, oh, that sounds really sweet, and then invite me to come and give lectures, and I'll talk about maggots for 60 minutes, which are some of the best baby animals in the world, and then I'll never be invited back again. But the idea is, what I want to get into this book, it's called Nursery Earth, Baby Animals and Why They Matter, and I want to talk about the fact that all animals, from cephalopods to humans to flies, go through this incredible part of our life cycle that really ties us to different habitats, to each other, to different species, because we all go through this part of our life cycle where we go down to being just one little cell, just a little fertilized egg cell, and then we have to build our body all over again. And that process involves meeting new symbionts, meeting new parasites, often living in a totally different habitat, even if you just think about like a caterpillar living on leaves versus a butterfly that can fly. And then with the cephalopods, we see it that so many of their eggs hatch out as these tiny little things called paralarvae that are plankton. They're basically drifters, and they just they live in the open ocean, and so they're the ones that are being eaten by open ocean things, and then they settle down into their rocky, denny habitat. And so it's really the babies that are kind of, I think of them as like little needles stitching together habitats all over the world. And I'm definitely going to talk about cephalopods in the book because I like their paralarvae a lot. But I don't know what to feed them. So what do you feed them, Rich?
You try anything. The one I worked with at home has kind of a benthic-ish baby. It's kind of like this mid-paralarva.
Benthic means sitting on the bottom.
Sitting on the. Thank you. You're so good at that.
Glossary.
But it kind of doesn't. Some of the other colder water octopuses do. They're very much their own thing. So the Chirchii, you feed them amphipods, but they also eat each other.
Right? That's actually the best thing to feed them.
Yeah, it is. I mean, I think that's what they're designed to do, just like humans. And
I think Taryn regrets asking this question.
Eat your siblings. So you have to keep them separated, which makes it harder. And when they're true paralarvae, out in the water column, they're growing so fast that they're eating. One day they'll be eating, this kind of larva from a crab. And the next day, they don't care about that anymore, and they're eating something else. And so trying to make that happen in a lab and anticipate and have that food stuff ready for them is daunting. I'm so glad Brett Grassie's at the MBL, and they're working on that. When they work with the Chirchii, they have a guy who cuts up amphipods and hand feeds each one 6 times a day.
Oh my goodness.
And that was the opposite of how I was trying to do it, because that's a lot. It's worse than my daughter. And
she only eats amphipods 3 times.
Yeah, 3 times a day, and she'll eat whole ones now. So it's that's the whole key. Is that there's like 50 whole keys. But that's one, what do they eat and when, and how do you get them to do it?
Okay, great question, Taryn, because we don't have the answer other than eating each other, which I'm sure you didn't really want to know. Josh, I'm going to ask the next question of you. It's a little bit of a two-part, but I saw my octopus teacher, sorry, I should just touch my microphone, as so many of us did. It was a big hit. Yeah. Was that a good thing, a bad thing that became so popular, the attention on cephalopods?
Seems like a good thing to me. I think I wasn't used to people, all the folks in my life that wanted to talk to me about octopuses on a regular basis. I think that was cool. It's a beautiful film. I don't know if everybody's seen it. But yeah, it's got, and also has that sort of personal aspect of like, a lot of when we like make films or write books or study things in nature, we're sort of thinking back about how it sort of either reflects something about ourselves or if there's some way that it sort of either affects us. And that's sort of in Deep Look episodes, we're always trying to think of like, how would, if I was a sea urchin larva that was swimming through the ocean, like what would that kind of be like, Not to necessarily give them like human emotions, but just to sort of, I don't know, I guess when we make films or write books about animals, we're also sort of learning about ourselves at the same time.
Sure.
I think of it a little bit almost as reverse anthropomorphism sometimes. Like we are inevitably putting our own imagination out there, but it also sort of stretches our minds to be like, what would it be like to be that? And I think it's
be right here.
Yeah.
In that film, we're meant to conclude that the octopus kind of recognized the filmmaker in some respects. And Rich, I'm wondering, you lived with these creatures. Like, are you under the impression that they recognized you?
Not only am I under the impression of that, but we know it to be true. My wife, Libby, always says, when you walk into the room and there's Cuddles there, they all rise up and come to the front of the tank. And shrimp come with people, people come with shrimp. They know.
Yeah.
But there was a study done with a giant Pacific octopus where one keeper would come in, they would dress the same. One keeper would come in and feed it. And another keeper would come in with a spiky brush later on. Not a spiky brush, a brush. It wasn't like stabby, it was just a brush. But the octopuses don't like that feeling and would interact with the octopus with that.
Yeah.
And very quickly, the octopus would react depending on who came in. If the feeder person came in, they would come close. If the spiky brush person came in, they would go hide.
Okay.
Yeah.
Great. Well, that was a question from Lydia online, so thank you very much for that, Lydia. Let's see here. We have a question from Harold Lee that I don't think we've asked and I've been wanting to. Are there any colors that we know that they cannot make?
Taupe.
It's not a color. Did you talk to folks about that in the lab?
I mean, I saw several different, there was like sort of reds and browns and yellows and things like that. There was also the iridophores and they making themselves more shiny or not. I don't know if some appear purple. Just I don't know the answer, but just from my observations, like there's a wide variety.
Certainly not every species can produce every color, and many of them are, especially the deep sea ones, often are very limited. But the chromatophores can do all the warm colors. They can do browns and blacks and oranges and yellows and reds, and then the iridophores, those reflecting cells, can do pretty much all the cool colors, greens and blues and purples. And then they have leucophores for white. I mean, and they can even make things that we can't see. They can produce polarized light that you could only notice if you had your sunglasses on and you tilted your head.
And I believe that each chromatophore can just do one color.
Yes, yeah, you really need the whole
like
system.
Pixels on your TV where it's only going to be, certain colors, but you can mix them together to make all the rainbow.
And then, of course, all that changes depending on how deep you are in the water column, because different wavelengths of light are absorbed by the water at different depths. So, yeah, they're wild.
Can you speak a little bit? We have an audience question about how do octopuses know what color to change their skin to? And I just find that fascinating. Can we speak a little bit to that?
They're so weird. So octopuses are colorblind. We have never found any way that their eyes can perceive wavelengths of light, which are color.
What about that chromatic aberration idea? Yeah,
it's an idea.
Yeah, okay.
I mean, it's a totally mathematically plausible idea that nobody has ever actually proven that they can do. So we have nobody has ever, and people have studied octopus eyes for a long time, and this is true, and now for all cephalopods, it's true for squid eyes as well and cuttlefish eyes, that we don't know, like our eyes have rods and cones, and we use our cones to distinguish between colors, different wavelengths, and octopus eyes, although in other ways they're very similar to our own, they don't have those different sensitive pigments. And so like this is one of the big mysteries. How do you match color when you're colorblind? And a lot of what they match is pattern. That's one aspect of it. And then, but there's clearly color matching too. And so the most going theory that I know of is that it comes down to the skin itself actually perceiving that, like the video was saying, like that it's as if you closed your eyes and your skin could tell what color was in the room or what color the walls were, but that the details of how that works and how that gets integrated into the color change system is still very
Fantastic. Great question.
The technical term.
This is kind of a big question, and we don't have a ton of time. There's one more that I really want to get to, but can we talk about the diversity, the speciation of cephalopods compared to other things that we have in the ocean? Like we just talked, I mean, you mentioned 5 of your favorites.
Yeah.
Right.
Yeah.
So, like, can we talk a little bit about, like, are they just exceptional that there are so many, or is this?
Well, I'm thinking about, how, it depends on how you classify it.
It does, because in terms of specifically numbers of species, the group cephalopods is not super speciose. It's got less than 1,000, I think.
Octopus, yeah.
Even cephalopods, totally.
Yeah,
I mean, maybe 1,000.
I think octopus around 400.
So then, if you compare that to something like snails, thousands and thousands of species of snails, or crabs. Oh my gosh. So, so in terms of numbers of species, they're not super prolific. in that regard. In terms of abundance, like the raw abundance of animals, there's crazy masses of squid in the ocean. And it's not so much that we know that because we'll go down scuba diving or send an ROV and run into like a thick mass of squid. It's because of the things that eat them. So like sperm whales, huge animals, live almost exclusively on squid. Same with albatrosses, they live almost exclusively on squid. So you can do the math and figure out that there's just like so many squid out there.
More cephs than fish by mass in the ocean is one of the estimates.
Wow. I actually did not know that.
Mark Norman, so
okay.
he said it, so we're allowed to say it.
Very good.
I think what fascinates me about just octopuses alone is people have this feeling that octopuses, if you talk about one, that they're all like that. And they're absolutely not. They're all incredible. They're not all, I'm over exaggerating, but there's so many different groups. You know, you've got the ones that live off our coast here in big aggregations we're discovering now, who might live 10 years, and you've got ones like the churchier that live in the tide pools and have bigger eggs, and they all need their own environment. It's not like you can go, this is how you keep octopus.
The Argonaut is an octopus.
Yes, the Argonaut is an octopus. This is how you keep this particular octopus, but the other ones are totally different. It's not, just the temperature difference is crazy.
There's so many more cephalopod movie nights and cephalopod weeks that are probably in our future as a result.
I hope so.
Well, with that, we're gonna wrap up the question portion. I'm sorry I didn't get to all of them. You had really excellent questions. Everybody will be in the lobby afterwards, so you can ask more questions then if you're here physically for the event. And Danna will be signing books. I've got some t-shirts and stickers and things like that. Some quick notes before I give you a little surprise, but there are flyers on your chairs so that you can get more involved in Cephalopod Week, like wallpaper for your phone. So please check it out, take it home with you. Okay, so we can't just leave you with conversation because we're here to watch things, and we have a very talented filmmaker here. So Josh, we have a movie or a short film that we're going to show right now as a sneak peek. Yeah.
So Deep Look, we recently celebrated our 150th episode. Woo! In our 9th season. And for this one, I made an episode about the surprisingly vigorous love lives of barnacles.
Okay. It's a great intro. So here we go.
Finding a date is hard enough in San Francisco, but it's especially tricky when you're stuck to a rock in the middle of the bay. Still, acorn barnacles don't get discouraged. These crusty little animals actually have a pretty wild sex life. At low tide, each one is sealed up inside its own miniature fortress, shielded by a ring of armored plates. The two central plates press together to form a watertight seal, so they don't dry out in the open air. They're ready and waiting for the tide to rise so they can get down to business. But first, they need to freshen up a bit. The barnacle unfurls 8 pairs of delicate feathery legs called cirri, which they use to absorb oxygen from the water. The legs filter out plankton and debris churned up by the waves, bringing the catch inside to the mouth. They may not look like it from the outside, but beneath their shell, it's easier to see that barnacles are crustaceans related to crabs and shrimp. After a nice meal, it's ready for some action. The little barnacle lets loose the longest penis of any animal, relative to its body size, of course, stretching up to 8 times the length of the barnacle itself. And this penis has skills. It can taste and smell, and the tip can feel around, probing to see which neighbors have ripe eggs inside. When it finds what it's looking for, the barnacle delivers sperm to fertilize the eggs. Barnacles aren't exactly prudes. Pretty much everyone is fair play, because they're all hermaphrodites, simultaneously male and female. Sometimes it's one-on-one, sometimes more. Barnacles are nurturing parents too. They hold on to their fertilized eggs and protect them until they hatch. These cuties are their baby larvae called nautilus. This is the young barnacle's chance for adventure. They roam the sea searching for food and growing. If they survive long enough, the barnacle larvae mature into cyprids. At this stage, the barnacle doesn't eat. The cyprid's only mission is to find the ideal spot to glom onto before it starves. Having survived the trials of youth, the barnacle settles in. Now it's time to get to know the neighbors.
That was a perfect way to end. So I want to thank our guests for joining us this evening.
Thank you.
Thank you.
I want to thank KQED and the Deep Look team, as well as the Science Friday team, who should largely be in bed. You can, once again, go to sciencefriday.com to listen to radio stories, watch some of our videos, read articles. You can find the same types of things at KQED, including the videos for Deep Look. We have some merchandise and some books to sign out in the lobby, so I hope that you'll join us there. We're also going to put a QR code up on the screen. We'll give you a Cephalopod sticker if you take a picture and fill it out. It should be very quick to do this. It helps tell us how we did, so we'd appreciate your help. And then lastly, rays of tentacles, if people would be willing to take a selfie for Science Friday.
A couple.
Hands up? Okay. Then I will, as soon as the lights come on, I will take a picture with all of you and you can put your tentacles up. So thank you very much, online audience as well, for joining us this evening. It's been a treat.
Thank you.